Hello guys, once again in this video we will continue from acid and base. We looked at acid and base, we started, and this video will continue from where we stopped on acid and base. So by the end of this lesson, the following success criteria shall be covered. The first one, we should be able to describe the uses of bases, state types of indicators, explain or describe how acid and base indicator can be prepared locally. So on the uses of bases, some of the common bases include the following. We have sodium hydroxide, we have calcium hydroxide, magnesium hydroxide, ammonium hydroxide, and potassium hydroxide. So in terms of the uses, we are going to work on each example that we have stated in the previous slide. So we are going to start with sodium hydroxide. So the first use of sodium hydroxide is that it is used in manufacturing of soap. It is used in petroleum refining. It is used in manufacturing medicine as well as papers. It is used in making rayon and textile industry. The second one is calcium hydroxide. It is also known as slag lime. It is used in manufacturing of bleach powder. It is used to neutralize acidity in water supplies. For example, in waterboard, they can use slag lime or calcium hydroxide to neutralize, if at all, to neutralize the acidity. In short, maybe to remove some salts in the water to make water soft. You can use slag lime. It is used usually mixed with sand and water to make a mortar. A mortar, in the next slides we'll be able to see how it looks like, but it is also used to make a mortar when it is mixed with sand and water. The next one is it is used to neutralize acidic soil in order to improve soil fertility. It is used as an antidote for food poisoning. It is used in preparing fungicides. The third base that we're going to look at is ammonium hydroxide. The following are the uses of ammonium hydroxide. It is used to remove ink spots from clothes. It is used to remove grease from window panes. It is used in water purification. In short, majority of ammonium hydroxide, it is used to remove stains when cleaning because it has bleaching property. Another one is potassium hydroxide. Potassium hydroxide, the following are the uses. It is used in manufacturing alkaline batteries. It is used to make soap as well as shaving cream, adjusting pH of the soil, to detect fungal infection on the skin, and to remove hair from animal hides. The last one that we're going to discuss is magnesium hydroxide. Magnesium hydroxide, it is used as a laxative or stimulation of bowel movement. It is used again as an antacid in treatment of heartburn. So those are the uses of bases which we discussed. Now from there we're going to look at in the acid and base indicators. So when we are talking about acid and base indicator, what is an indicator? So an indicator can be defined as follows. It is a special substance that is used to test or identify whether a given substance is an acid or a base or neutral substance. So an indicator helps someone to know if the solution which he or she is working with is a base or is a neutral substance or is an acid. So acid-base indicator will change to a certain color in acid solution and a different color in a base solution. What does this statement mean? It means that when you have an indicator, it shows variation in colors. It doesn't show the same color when you put it in either an acid or a base, but it shows different colors when it is the same indicator is added to a solution of a base or an acid. So the acid-base indicator to be tested must be always in a solution form. This means when you have a solution which you are suspecting, maybe you are working in the lab or the solutions are colorless, you cannot tell that this one is an acid or this one is a base. So what you're supposed to do is to use an indicator, because an indicator shows variation of different colors when it is added into different solutions. So we're saying that the solutions which are supposed to be tested must always be in liquid form. That's why the statement is saying the acid base to be tested must always be in a solution form. This tells you that you cannot use a solid substance to test it as either acidic or basic. For example, you cannot take direct soil and use an indicator to know if the soil is acidic or basic. It must always be in solution form. So there are two types of acid-base indicator. We have simple acid-base indicator, as well as we have commercial acid-base indicator. So we are going to look into the definitions of these ones and their differences. So a simple acid-base indicator. It is if we look at this word simple, simple tells us that firstly it can be easily made. Also simple can say that it can be easily found. So when we're looking at simple acid-base indicators, these are indicators obtained from direct plant leaves extract. So the word simple, it tells us that since the material which we can use to make the indicator is locally available, that's why simple, that's why the name becomes simple acid-base indicator. So preparation of this simple acid-base indicator, since the materials which we can use, they are locally available, so we are going to look at how we can prepare a simple acid-base indicator. So we have a question here, a sample of a question, how it can be asked. Describe how you can prepare acid and base indicator. So we have a question at hand. So the aim of our experiment, it will be to prepare a simple acid-base indicator, and the materials that we need a mortar and a paste, we need propanol or ethanol, we need a funnel, we need tomato leaves or hibiscus flowers, as well as other flowers that can be available. We need beaker, we need a cloth or filter paper. So from here we're going to look at the procedures that we follow when we are preparing a simple acid-base indicator. Remember, we're saying a simple acid-base indicator, it can be an extract from flowers. So the procedure, first of all, we need to take some of the tomato leaves and put them in a mortar. A mortar is this one. This one is a mortar, and a paste is this one. So we are going to collect tomato leaves and plug them properly and put them in a mortar. From there, you're going to crush using the paste. You're going to crush the leaves using the paste. Add little amount of propanol or ethanol. Why do we add little amount of propanol or ethanol when we are preparing the simple acid-base indicator? Remember, we are talking about an extract. It is the extract from flowers is the one which we are going to use, and flowers have a strong cell wall. It is not easy to get the extract from flowers, so we add propanol or ethanol just to break the cell wall in order to get the extract as we are crushing the leaves. So After adding propanol and ethanol, grind the leaves until you get enough extract on the flowers. So it means you still crush, continue crushing and grinding it until you get enough extract from flowers. Using a piece of cloth now, if you know that now I have enough extract from your mortar, it means you are going to use a piece of cloth or filter paper. We have put these two. Sometimes it is very hard to find filter paper, but the same thing can also be done using a cloth. Filter the extract into a clean beaker. You are going to get the extract. It means you're going to remove some of the materials which were there. Note the color of the filtrate. Here it's very important to observe this. You need to note the color of filtrate. The addition of propanol or ethanol may change the complexion in terms of how the color of your extract look like. So if it is faint, it means you need to boil. If you boil, you are going to remove some of the solvent that you used to remain with the actual extract. The remaining mixture after boiling is an indicator which is known as a simple acid-base indicator, which can be used. Now, to use that particular indicator, it doesn't mean that when you have extracted it, you are done. You need to test if it is really working. Remember, we said an indicator is a substance which is able to identify if the solution is either an acid or a base or a neutral substance. So usually in the simple acid-base indicator, you will be able to know that this one is an acid or a base. So how to use the indicator that have been extracted from flower leaves or tomato leaves. First of all, you measure reasonable amount. Here we're not talking about reasonable amount. It can be 5 mils, whatever mils that you want, and put it measure reasonable amount of the solutions that you want to test. For example, in this case, we want to test soda solution, hydrochloric acid, lemon juice, toothpaste solution, sour, milk, and pour it in different beakers. So after that, you're going to take, using a dropper, you are going to add 2 to 3 drops of prepared indicator in the test tube containing the solutions that you want to test. Record the color change in the table below. So we have a table here. Since you are trying to check if your indicator is working, so you have the substance, so you'll be able to record the color which is changing after you have added the indicator. So you will record and be able to tell that this one is either an acid or a base. Then the next type of indicator is commercial acid-base indicator. These are indicators that are prepared in industries. Usually, they are found in laboratories. It can be school lab, it can be in the hospitals. We will have these prepared indicators. Examples of these commercial acid and base indicators include the following. We may have a solid one, that is litmus paper. We have phenolphthalein indicator, methyl orange, screen methyl orange, and universal indicator. Amongst these ones, usually in school labs, you may find there's a litmus paper, phenolphthalein indicator, as well as a universal indicator. So let us go into more details about these indicators. So color change when different indicators are added to acid and bases. So we have first of all litmus paper. Litmus paper, we are going to look at litmus paper into details, but litmus paper usually is an absorbent paper. It can be of different colors, but even if it is of different colors, but it will always show the same results when it is dipped in an acid as well as a base. So first of all, if you have litmus paper and dip it in an acidic solution, we expect that one to change to red. But when we dip it in alkaline solution, we expect to change it to blue. Methyl orange, when you add the drops of methyl orange into an acid, the color changes to pink, while when you add methyl orange into an alkaline or base, the color changes to yellow. Phenolphthalein indicator, when you add it to an acid, the color becomes colorless, while when you add it into a base, the color changes to pink. The screen methyl orange, when you add it in a acid, becomes purple, while in a base becomes orange. From here, we did not include universal indicator. There is a reason why in this table it is not having universal indicator. We are going to discuss it more in the next slides. So here we have the colors. For example, color change of litmus paper. So you can see this is a litmus paper, and here it's a blue litmus paper. But after you have dipped it in an acid, it is changing to red color. Well, this one, it's a red litmus paper. After you dip it in a base, you see it is changing to blue color. The color change of methyl orange in acid and base solution. So this is methyl orange. You can see the label. And if you add it to an acid, you have pink, while when you add it to a base or alkaline, it's yellow in color. Then this one is phenolphthalein indicator. When you add phenolphthalein indicator in an acid, the color becomes colorless, while in a base, it's pink color. Now, commonly found commercial indicators in school laboratory settings. So, as I said in the previous slides, I said that most commonly found commercial indicators in school laboratory, we have litmus paper, phenolphthalein indicator, as well as universal indicator. So we're going to look into details into these commonly found commercial indicators. The first one is litmus paper. So what is a litmus paper? A litmus paper is an absorbent paper which has been dipped in litmus indicator solution and dried. So that paper is just prepared. It's not a normal paper. It's a paper that is going to be dipped and later on dried. After being dried, it is used as a litmus paper. But we're going to look at how it is prepared. So in this statement, saying litmus paper is a blue vegetable compound which is extracted from plants called lichens. So here we have a diagram. When we're talking about lichens, these are the lichens which we're talking about. Mostly they are found in natural trees, usually in a forest. They are there, so you can take them and extract some litmus indicator from it. Then you can use a paper, an absorbent paper, and dip it in the solution that is going to be prepared and form a litmus paper. So litmus paper can be found in different colors, but here we only have two colors. We have blue as well as red, but it can also be found in pink, orange. We also have colors of such litmus papers, but the way they work in the same way. If it is dipped in an acid, depending on any color, it changes to red, but if it is dipped in a base, changes to blue. How to use litmus paper? So first of all, you put reasonable amount of the solution to be tested in different beakers. Dip either blue or red stripe of litmus paper or of any color and observe the color change. If it changes the color to blue, it means the solution is a base, but if it changes to red, it means the solution under tested is an acid. So we can see here, if this is an acid, the color change of litmus paper is red, but if it is a base, The color change on lead litmus paper is blue. Universal indicator. So universal indicator, it is a mixture of dyes. There are many dyes which are used to make universal indicator. It gives a range of colors depending on the strength of an acid or base. The beauty of universal indicator is able to tell you that this acid is strong or weak. That's why we are saying that it gives ranges of colors depending on the strength of acid or base. Universal indicator works together with pH, standard pH color chart, to determine the pH of the solution. Later on in the next lessons, we'll be able to see how this works. How does the universal indicator and the pH scale work together to determine the strength of an acid or a base? So here we have a bar session. The word bar session simply means it's a quick exercise that we need to attempt. First of all, the first question is state two uses of sodium hydroxide. So we can usually get the uses of sodium hydroxide. Remember, sodium hydroxide is a base. We have many uses of sodium hydroxide. The question number 2, table 1 shows pH values of substance P, Q, R, S, and T. So we have P, Q, R, S, T, and here we have the pH values. So the table is going to help us to answer questions. So the first question, what would be the color of litmus paper if it is dipped in the substance T? So you'll be able to know if T is an acid or a base, so then you can be able to answer this question. The next one is choose a pair of substances which will likely form a neutral solution when they react. You use the same table to answer this question. Question number 4, a laboratory assistant at St. Mary's Girls Secondary School prepare sodium hydroxide solution and sulfuric acid in separate beakers but forgot to label them. Explain what he should do to distinguish them. That's question number 4. So you can use the concept discussed to answer the question number 4. Question number 5, describe how acid-base indicator can be prepared locally from hibiscus flowers. So we can usually describe this experiment. Now, if there is any question, we have a WhatsApp number which you can follow up, as well as you can visit Xulendo for more information. But so far, we have covered the uses of different acids and bases. We have defined an indicator as a substance which is used to identify if either a solution is a base, acid, or neutral. We have looked at types of indicators. We have simple acid-base indicator as well as commercial indicator. The difference between them is that simple acid-base indicator can be made from plant material by getting an extract, while commercial is prepared in the lab. So if you were to ask me further details, simple acid-base indicator cannot stay for long period of time because it doesn't have some preservative. So while a commercial one, it stays for many times in the lab. It can be stayed over for 1 year, depending on the lifespan of the preservative solutions that we use, but it can stay more than that because it has some preservative material in it. So examples of commercial indicator, we have mentioned them, as well as the most common commercial indicator that can be found in the lab. We have mentioned 3. We have phenolphthalein indicator, we have universal indicator, as well as litmus paper. So this marks the end of the presentation of the concept that we are supposed to discuss. So if there is any further research that you would like to make, you can use the references, the following references: Excel and Succeed Junior Chemistry Book 2, Achievers in Chemistry Book 2, Target Junior Chemistry Book 2, and the photos that we have used here. On the photos that we have used here, these are the links that we can use to get the photos. This marks the end of this presentation.